Bioinfocongress. Dr. Pınar Zorlutuna. Engineered Disease Models with Aged Tissue. Lisans eğitimini Ankara Üniversitesi Biyoloji bölümünde, yüksek lisans eğitimini Orta Doğu Teknik Üniversitesi Biyoteknoloji bölümünde tamamlamıştır. Doktora derecesini Orta Doğu Teknik Üniversitesi ile Londra Queen Mary Üniversitesi Biyomedikal Malzemeler Disiplinlerarası Araştırma Merkezi arasında ortak bir projeden almıştır. Biyomalzemeler ve doku mühendisliği ile ilgili 2 patentin ortak sahibi olan Dr. Zorlutuna, araştırmalarında doku mühendisliği, genetik mühendisliği, mikro ve nanofabrikasyon yaklaşımlarını kullanarak hücre davranışlarının çevreyle etkileşimlerini anlamak için biyomimetrik ortamlar tasarlamayı amaçlamaktadır. Şu anda Notre Dame Üniversitesi'nde profesör doktor olarak kurmuş olduğu Zorlutuna laboratuvarında çalışmalarını sürdürmektedir.
Sayın Dr. Pınar Zorlutuna hocamız ve değerli katılımcılarınız, kongremize hepiniz hoş geldiniz. Değerli hocamız bizlere bugün Engineered Disease Models with Aged Tissue Microenvironments konulu bir konuşma gerçekleştirecektir. Konuşma bitiminde ekibimizin hazırladığı ve katılımcılardan gelen soruları hocamıza sunacağız. Tekrardan hoş geldiniz hocam.
Merhaba.
Nasılsınız hocam?
İyiyim. Çok teşekkür ederim. Çok teşekkürler davetiniz için.
Sağ olun hocam. Dilerseniz sunumunuzla başlayabilirsiniz.
Pekala. Tam ekran yapayım slaytlarımı. Görebiliyor musunuz slaytları?
Evet hocam.
Tamam. Çok teşekkür ediyorum. Ben Türkçe konuşma vermeye alışık olmadığım için ne yazık ki İngilizce vereceğim bu konuşmayı izleyicilerden izin alarak. Çünkü en sevmediğim şey yarı İngilizce yarı Türkçe konuşmak. Onu yapmak istemiyorum. O yüzden konuşmaları İngilizce vereceğim ve soruları Türkçe cevaplamaktan memnuniyet duyarım. So today's talk is about engineered disease models with aged tissue environments, but I would like to first introduce my lab at University of Notre Dame because we work on several other research projects that may be interesting for the audience of this talk. We have 2 research trusts, and the first research trust is on cell-based robotics and computing. In this research trust, we investigate using live cells, especially cardiomyocytes and cardiac fibroblasts, as computing elements. So we study their properties in terms of electrical signal propagation, and we have in the past used microfabrication methods to create platforms that have that has the ability to propagate signals in a directional manner. Here you see what we call a living diode. So a diode, for those of you who are not familiar with the terminology, is an electrical circuit element that allows a signal to pass from one side to the other while not allowing the reverse signal to pass. And that's one of the basic elements for creating circuitries for computing applications. And recently, one of the areas that are studied, investigated under new computing paradigms are using bioelements for computation. And we investigate cardiac cells for this purpose. Here you see a cell-based diode that is created by using cardiac muscle cells on one side and the cardiac fibroblasts on the other side. Cardiac muscle cells are excitable cells. Once they receive a signal from the cardiac end, the signal passes through the interface and then still can pass through the fibroblasts and be collected on the other end. So the fibroblasts act as a passive conduit, but still allow the signal to pass. On the other hand, if the same signal comes from the fibroblast side, it cannot propagate beyond the muscle boundary because fibroblasts cannot amplify the signal that they receive. So this was a proof of concept that these cardiac cells can be used as computing elements, but after that initial study, we have started to investigate the other possible usage of these cells as computing elements, and we have conducted studies where we use the cardiac cells as biooscillators and as elements for collective computing. So all of these cardiac cell-based biocomputing circuitries that we are working on can potentially be integrated with biorobotics. But for that to happen, we also work on technologies using nanoparticles and novel sensors that can capture the membrane potential changes of these cells in subcellular scale. And also we work on technologies to integrate these muscle cells to devices. And you see here a little biobot that swims in the interface of a cell culture media flask. And the tail here is propelled by the beating activity of the cardiac muscle cells that are seated on it. So all these technologies can be potentially integrated with biorobots that are produced by micro nanofabrication methods and that are actuated by the beating or actuation of muscle cells. On the other hand, on the more biomedical side of things, our second research trust is on engineered tissue and disease models. And then in this research trust, we are very much interested in how cells are interacting with their extracellular matrix, and then how cell extracellular matrix interactions define the healthy and disease state. And we also look into how stem cell differentiation is affected, especially, again, with respect to the stem cells' relation to their extracellular matrix. In both these projects or research areas, we are very interested specifically looking into what changes in aged tissues and then how aging ECM affects either the healthy and disease state of the tissue or the differentiation state of stem cells. Especially in both projects, we are interested in using these models that we create. I mean, on one hand, we study the basic science and how these interactions affect and alter the disease state. But we are an engineering lab, and then we make use of those models to study applications related to these diseases. And in one example here, we look into how organ preservation can be achieved better using stem cell-derived products. And we assess the effectiveness of those products using heart-on-a-chip technologies that we use creating microfluidic devices and stem cells, specifically induced pluripotent stem cells, derived cardiomyocyte and endothelial cells. We also use these disease models for novel biomarker discovery, and we look into how we can come up with markers that are potentially faster to detect diseases. And we look into also, in conjunction with this study, we look into novel sensor platforms that can be used for faster biomarker readings from either plasma or tissue samples. And today's talk will be about this part here, cell ECM interactions, and how the aged extracellular matrix affects those interactions and potentially drive healthy cells to a more diseased state. But first of all, I would like to motivate the need for engineered tissue models. So this is a very currently hot topic / very investigated, very highly investigated field under bioengineering, and there's a good reason for that. Almost 90% of all drugs that are prone to be effective in preclinical models that use mostly mouse or rat, small animal models, prone to be not really effective for clinical treatments. So only 10%, in other words, only 10% of drugs that show effectiveness in mouse models pass the clinical trials once they started to be investigated using human subjects. And there's a good reason for that. Mouse and men are not the same. So this graph here from a publication earlier, a few years ago, published in the journal Nature by a leading group that compared the mouse and human genomes, show how much the genes in each organ is preserved. So here you see the human organ and the mouse organ. Human organs are shown in solid squares, and the mouse organ is shown in empty squares. And you see that human organs cluster together if you look into the whole expression patterns of these organs, and then the mouse organs cluster together. What it means is the mouse, for example, if we look into mouse intestine, which is right here, is totally uncorrelated with the human intestine, which is here. And if you look at it, the human kidney is more close to human intestine than mouse intestine. So in other words, if you're studying a certain cancer type, you may be better off looking into other cancer types in human than the cancers occur in mouse on that specific tissue. And there's a reason for that. So although evolutionarily many of the intercellular organelle level genes are conserved, like here's a graph showing that. So nuclear, intracellular, and organelle level genes are conserved across different species, but especially processes or structures that are related to cell-cell and cell extracellular matrix interactions. So things that are beyond just inside the cell, like vasculature, motility, cell adhesion, immune system, and so on. So all these more interaction-related processes in the organisms are not very well preserved in between different species, especially in this case, mouse and human. So that's why bioengineers have been motivating using the engineered tissue models that constitute of human cells as an alternative method, not to maybe totally replace mouse models, but a model that should be taken seriously and used alongside the mouse models to give the human relevance, while the mouse model can be used to look into more systemic effects. That's why engineered tissue models have gained a lot of attraction across the world in bioengineering labs. And our lab have been working on creating age-relevant tissue models for studying diseases. And one of the reasons I believe that low translation rate of preclinical studies to clinical studies, in addition to being mouse and human being different, is the lack of age-relevant models in the preclinical studies. Most of the mouse models for diseases that oftentimes affect the older population. In this case, you see a graph here that shows the population distribution of breast cancer incidence. And breast cancer happens mostly in older age. The median age is around 65 here, while most of the studies, preclinical studies to either understand or treat breast cancer are done using 3-month-old mouse, which is equivalent to 25-year-old human. And that's a big discrepancy. There's a huge gap in the clinical studies with respect to what's happening by age and how it may be altering the disease state. And this is true for cardiac diseases too. And in this talk today, I'm going to talk about our studies around the idea of how the aged extracellular matrix in breast cancer is affecting the healthy epithelial cells and how they may be driving them to a more diseased phenotype. But on the other hand, on other projects in my lab, we also look into heart disease equivalence of this idea, where we study what's happening in the heart extracellular matrix as it ages and how it may be contributing to the lower degree of healing that happens in an infarcted heart at later ages. So obviously aging is a area that has been under extensive research, but many of these studies look into how cell-level changes are affecting the potential disease state of the cell, like telomere shortening, mutations, impaired repair mechanisms, impaired immune surveillance that happens at older ages, which all of it is, I believe, true to be contributing to the higher occurrence of these diseases at later age. But also, in addition to all these changes, your extracellular matrix changes by age. And so far, no study have focused on what's changing at ECM as you age and how it may be directly contributing to the disease state of the cells. So there is a increased appreciation of how extracellular matrix alone affects the cell behavior. Again, not in the context of aging, but there are other studies that looked into the influence of extracellular matrix properties, for example, in occurrence rates or in the aggressiveness of cancer. And people have shown that both the biophysical and the biochemical properties of extracellular matrix affects the cancer cell occurrence and progression. It's like now well accepted, well appreciated that there's this analogy of seed and soil. So if the cancer is the seed, it needs the microenvironment, which is the soil, and it alters the soil, and that's how it becomes aggressive and metastatic and so on. And studies investigate how this change in the stiffness, pore size, and organization and alignment of the fibers may be affecting the cancer occurrence and or progression. So, our hypothesis was, when we first started this project, is that aging ECM changes in terms of multiple of these properties, and it may be supporting cancerous phenotype. So our hypothesis was aging ECM supports cancerous phenotype, and that's one of the reasons why you see increased cancer incidence by age. So in order to test this hypothesis, we started to investigate this using young and aged mouse tissues that we extract from 3 to 6 month old mouse, which is the current preclinical gold standard that is used in the studies, in many studies around the world. So it is 20 to 30 years age equivalent to human, and then 20 to 23 month old aged mouse, which is 60 to 67 years of age in human years. And we wanted to first study what is happening in these tissues as aging occurs, how the extracellular matrix is changing. And this study was mostly conducted by my postdoctoral researcher, Gökhan Bahçecioğlu. So I would like to acknowledge him, because that's mostly his hard work that we discovered very interesting very interesting ideas around this concept. When we first started looking into what's changing in ECM of aged breast tissue, there was almost no literature on that. So we first started by very rigorously characterizing the changes that are happening in the native tissue itself. So we started with the native tissue. We use a gentile decellularization process and then a delipidization process to get rid of the fat around the breast tissue, because it's a very fatty tissue. You need to have an extra step to get rid of the fat. And then we characterize the tissues, comparing the native decellularized and the lipidized ones, to ensure that we are preserving the extracellular matrix during this process. And then after thoroughly characterization all these both the biochemical and biophysical changes that are happening in these tissues, we seeded these cells with either normal, totally healthy breast epithelial cells or cancerous breast epithelial cells, and looked into cell behavior in terms of their normal spheroid formation and migration patterns and gene and protein expression levels. So this is a bit of a busy slide, but I'm going to walk you through this. As I said, we wanted to understand first what's changing in the structure and composition of the breast tissue matrix as it ages. And this is, again, totally healthy breast tissue. There's no tumor in any of these animals. And on here on the top left, you see the second harmonic imaging of these tissues. Second harmonic imaging shows the collagen fiber structure. And first thing we noticed, which was really striking, is that the young tissue has thicker fibers in the extracellular matrix, so thicker and more straight bundles of fibers, while the aged tissue has more curvy, thinner but denser fibers in their extracellular matrix. And when we characterize this through multiple tissues and sections and multiple different spots on the tissue, we saw that the fiber diameter is significantly less in the native tissue, and we because we did the decellularization and delipidization process, we always compared it to the native one, but all the tissues we looked into, native, decellularized, and delipidized, showed the same trend. So the aged tissue has thinner but more curly fibers. Then we looked at its mechanical properties on the other hand. Using a colloidal probe and a nanoindenter, we saw that the native tissue, because of the high fat content, at the first glance looks like it is softer for the aged tissue. But once you get rid of that fat, you start to see that the aged tissue has actually a higher Young's modulus, which agrees with these images that shows denser but thinner fibers. And then we looked into what's happening in terms of the content of these ECMs, and we saw that the biochemical composition changes significantly. Specifically, collagen 5 and 15, which are very important in collagen fibril assembly and attachment of the basement membrane to the stroma in breast tissue, are downregulated in the aged ECM. At the same time, collagen 1 is much more pronounced in the aged breast tissue. And then when we look at the cytokine content of this extracellular matrix, we saw that there's an increase in the pro-inflammatory cytokines and cytokines that may favor EMT and transition and metastasis. So after looking into all these and investigating all these changes that are happening between young and aged tissue matrix, we seeded these decellularized and delipidized matrices with healthy epithelial cells that are from healthy donors. And these are human cells, by the way. We seeded the cells on the young and aged matrix, and we started to culture them, and they started to form these nice acini-like structures, because these are epithelial cells, primary healthy epithelial cells. They should be forming structures like these, and the spheres that they're forming should be really round and nice and homogeneous. And we saw that in the young matrix, cells seeded on the young matrix. But when we looked at the cell phenotype on the aged matrix, we first saw that the spheres that the cells form on the aged matrix are not as nicely rounded, so they are elongated like a football shape. And then when we looked into it from a quantitative point of view and compared different samples and sections, we looked like the circularity is much less in the aged cells on the aged tissue, and the aspect ratio was higher, confirming these images that we observed in the microscopy. So more interestingly, after a certain time, which is in our culture was day 14, we started to see those spheres disintegrating on the aged matrix. So the young matrix still harbored these nice spheres, even on day 15 and beyond, actually. But the aged matrix couldn't support the healthy sphere structure beyond 2 weeks. And these first elongated spheres started to disintegrate, and the spheres are lost on the aged matrix. So this was very surprising, because these are totally healthy cells that otherwise coming from the same source, but acting very differently based on the matrix age. And the top image shows the bright field images of these spheres, and the bottom one shows, again, the second harmonic, which is very nice because you can both see the collagen fibers that are thicker in these young matrices and the dense and thinner and curlier fibers in the aged one. So we started to think what may be the reason, and obviously there are two options. It could be weak attachment of the cells to the matrix or degradation of the matrix by the enzymes. So we decided to further look into this by using some immunostaining, and then we checked for cell-cell adhesion proteins, in this case E-cadherin, which is the major cell surface protein that governs the attachment of these cells to each other. And it's supposed to mostly be on the cell periphery, on healthy epithelial cells, which is shown in this figure on the cells on the young matrix, were localized in the interior of the cell and disrupted from the periphery on the cells on the aged matrix. So the cells on the aged matrix didn't have those cell surface proteins on the right spot for the cells to form proper cell-cell interactions and form the sphere structure that was intact on the young matrix. Similarly, we saw an increase in the MMP2 production, which is a enzyme that degrades the extracellular matrix from the cells that are cultured on the aged matrix, and collagen 1 production was also increased. Then we were curious, if the healthy cells are behaving that differently, how would the cancer cells behave on these young and aged tissue matrices that are otherwise healthy? And we took time-lapse images of these cancer cells that are tagged with green fluorescence protein, and the cells were much more motile on the aged tissue, as you can see here. And also you'll see some dividing cells even in the short time period of recording that we conducted in this study. It is like 24 hours. And again, this is a image that were taken by like a two-channel in a two-photon confocal, with second harmonic being one of the channels. So the fluorescence is overlaid and showing the green cells, but you can also see the fiber structure. So the red is the collagen fibers of the tissue, and the young one shows the nice, dense, thicker fibers that are not as permissive for the cells to become really like jiggly around this whole tissue, and the aged matrix shows the dense and curlier fibers here. So when we quantified this behavior, we saw that that observation that I just showed on the video, which was already very striking, is quantitatively reproducibly the same in different samples we tested, and the aged tissue promotes a much more motile cancer cell phenotype. These are NDA cells now, the cancer cells that are being much more motile on this tissues that are from the aged animal. And then we wanted to see how the normal cells would behave, and then see that the same matrices with this time the normal epithelial cells, and saw the same trend. The normal epithelial cells were much more motile on the aged matrix. And also we looked into, this is like 2D motility, right? The cells are moving on the XY plane, but we wanted to see how invasive these cells become after being seated on these tissues. And then we did a 3D invasion assay, and we saw that the cells become much more invasive, in addition to being much more motile, as shown here. And we wanted to look into, at a genetic level, what may be the reason for this. Because obviously we see the changes in the extracellular matrix through the biophysical and biochemical characterizations we have done, but what's changing inside the cell based on those changes in the extracellular matrix that is driving a more motile, more invasive phenotype for otherwise healthy epithelial cells. So we see that again, once again, these healthy epithelial cells on our matrices, and the single cell sequencing and looked into what's changing in terms of the gene expression patterns in these cells that are grown on young or aged matrices. So at the first glance, it looks like there are similar populations, and it is expected, and this is a particularly healthy epithelial cell, single cell transcriptome analysis outcome, which is great because showing that we are dealing with healthy epithelial cells here. But then if you look into the clusters and analyze it, and this graph on the left shows the global gene expression, and the clusters show the single cell level. And both at the single cell level and at the global level, I'll start with the global level. So it was very interesting to see the cells on the age matrix were upregulated for some of the genes that are associated already with breast cancer. So we saw an upregulation of genes that are related with E-cadherin loss and invasiveness and EMT transition and breast cancer, and downregulation of the opposites. And we actually, when we did the single cell analysis, we identified 9 clusters. And on the global level, there were 29 genes that are upregulated compared to the young matrix on the cells on the age matrix. And many of them are, as I said, associated with the genes that are relevant to EMT and invasiveness. And I'm going to specifically talk about one of those in the next few slides. But first, I want to emphasize, when we did the single cell level analysis and clustered these cells, we saw that especially the cluster 7 cells were enriched for certain genes. So this global increase in certain cancer-related genes, if you look at single cell level, you'll see that that increase mostly being contributed by a subset of these cells that start to produce these genes at a much more higher level. So in other words, there's a subpopulation of these healthy epithelial cells that become more cancer-like just because they are being exposed to this age matrix. And there are, as I said, many cells, many genes that are like 29 of them. But the most highly expressed one in this cluster 7 that we identified as a cluster that starts to change was LOX, lysyl oxidase. And that's very interesting because lysyl oxidase is an enzyme that cross-links collagen. So it is an extracellular matrix related enzyme. So we decided to go after this change in LOX, and we used siRNA to downregulate LOX on cells seated on young and aged matrix. And very interestingly, the phenotype of the cells, of these healthy cells, that shifted in the age matrix to a more invasive, more EMT-like phenotype by the loss of E-cadherin and the loss of the spheres, was reversed. So in the young matrix, both with the control scrambled siRNA and the LOX siRNA, we saw the same trend of nice E-cadherin localization at the cell periphery and the sphere preservation. But when we repeated the experiment with the cells on the age matrix, the control group with scrambled siRNA ended up again losing the E-cadherin expression, and the spheres were lost beyond 2 weeks. But we saw a rescue pattern in the cells that are treated with siRNA for LOX in the cells that are seated on age matrix. Spheres were there beyond 2 weeks, and the ECAT was back to cell periphery. So this was very exciting, and we looked into what we would get in terms of the motility invasion behavior we observed, and we saw a similar rescue pattern for the cells that are treated with LOX siRNA. The increased motility in the age matrix was reversed back to normal cell motility levels, and the invasiveness also was reversed back on the aged tissue for after treating with LOX siRNA. We saw a similar trend for the cancer cells, motility and invasion. So the more motile, more invasive phenotype was reversed back after the treatment with siRNA for LOX. So as a summary of this part, I would like to say that on the aged ECM, the deformation and disruption of the healthy epithelial spheroids seem to be governed by E-cadherin delocalization and increase in MMP2 levels. Upon single-cell gene expression analysis, we saw several genes that may be contributing to that, which we started to investigate, but the lysyl oxidase, or LOX for short, seemed to be one of the leading players on this. And when we knocked down that gene, we were able to reverse most of the aging-associated changes that we saw on the cells that are grown on aged matrices. And there may be many reasons for that. The decreased collagen 5, collagen 15, and increased chemokines, and increased stiffness and collagen density, altered collagen structure. So although we identified LOX as a changing key factor, and knocking down it reverses the changes associated with this aging-associated changes, we still don't know which aging-associated change really causes this LOX increase. As going back to this first slide, there are several things that are changing: the ECM structure, the composition, the stiffness, the cytokine content. All of them are changing. So in the breast cancer microenvironment, similarly, there is like a lot of parameters that are changing. So this brings me to the second and shorter part of my talk, which motivates this basic science knowledge that we obtain from studying the native tissue and how cells are interacting with their native tissue, and how we can turn it and use it in our engineered disease models in order to identify new targets for cancer therapy. So on other side of things, as I said, once we study and understand what's happening in the native tissue in healthy and disease and aging state in this specific case, we try to recreate such tissues in the lab using tissue engineering techniques, and then we study those factors parametrically. As I said, there were like several things changing in the aged breast ECM. We don't know which one or which multiple ones of these factors are affecting the change we observed in the LOX expression, eventually leading to more cancer-like phenotype for normal epithelial cells. So these engineered tissue models allow one to have tunable stiffness, flexible extracellular matrix components, and you can define the tumor size, and you can incorporate different stromal cells. In this study that I'm going to talk in the last part of my talk, we looked specifically into the influence of adipocytes, and you can potentially use human cells and human relevant matrix components and have an engineered tissue model that can be used for parametrically studying the potential influences in the aged tissue microenvironments. And in this study, we were very interested in one of these factors, stiffness particularly, and we wanted to create a platform, a flexible platform that we can study the interaction of stromal cells with matrix stiffness, because matrix stiffness is known to be contributing to cancer disease state, but there was little known how much the interaction of the cancer that the surrounding stromal cells. In breast cancer, it is mostly adipocytes, fat cells, how those interactions are governed by the matrix stiffness. So in order to study this, we created this engineered disease model where we have a microfabricated array of microvalves that defines our tumor size. In this top view, you see the cells confined in these little valves. These are cancer cells, and then in the microvalve itself, which is made of a hydrogel that we can tune the stiffness of, adipocyte progenitors are embedded. So how we fabricate this is we have a hydrogel solution that we can vary the stiffness of by tuning its chemistry, and then we incorporate progenitors for adipocytes in this hydrogel mixture. And then we put it on a stage where we can use a microfabricated stamp with microvalves and shine a little bit of UV light to cross-link, which traps these progenitors in the body of the microvalve array. And this creates a tiny array of little valves for the cancer cells to grow and form spheroids. So once the adipocyte containing microvalve array is ready, we seed the array with the cancer cells and then look into interactions of the cancer and the adipocytes. And in order to tune the stiffness of these, we used metacrelated gelatin, and we looked into 3 different stiffnesses, low, medium, and high, corresponding to healthy and diseased breast ECM stiffnesses. So, and then once we encapsulate these adipocytes in this hydrogel material with different stiffnesses, we start the differentiation process because these are progenitors. And then at one point of the differentiation, once the initial differentiation is initiated, I would say, we seed the microvalves with cancer cells and then look into what's happening to the cancer and the adipocytes around the cancer. And this is part of a larger study, but in the interest of time, I will just show one very striking result of this, highlighting the point that parametrically studying what's going on in a tissue is very possible using engineered disease models. So I will start with the observation we had after we started to use this model. We looked into differentiation degree of these adipocyte progenitors to fully grown adipocytes. So this staining here shows cell nuclei for blue and red for adipocytes, because it is a nilaret staining that stains the fat in adipocytes. So here you see the control. Remember, metacrelated gelatin low is for healthy breast stiffness, which is around 0.5 kilopascals. And then JELMA high is for metacrelated gelatin high for breast cancer-like stiffness, which is around 1.6 kilopascals or so. So when we had the control valves that have the progenitors that are seeded and the differentiation completed without without the tumor spheres being present, the differentiation process is really smooth, and then you end up having a good degree of adipocytes differentiated from these progenitors. But then in the presence of the cancer cells, and we tried this with 2 different cell types, HCC and MDA, both are invasive human cancer cell lines, we saw that there's a decrease in the adipocyte differentiation, which is controlled by the presence of the tumor spheroids. So when you have the tumor spheroids grown, the differentiation degree was lower, as you see with the less red staining here. And this observation led us to ask several questions. So the phenomenon we observed was adipogenesis was inhibited by co-culturing with cancer cells, but in an ECM stiffness-dependent manner. And there are 4 possible reasons we thought about that could lead to that. So cancer cells may be secreting factors that have effects on adipogenesis only when matrix is stiff. And in this scenario, adipose progenitors are the ones that are sensing the stiffness. A second option would be cancer cells secrete factors for interrupting the adipogenesis only when the matrix is stiff. So in other words, the cancer cells sense the stiffness, and they produce factors only when the matrix is stiff. There could be a third option where adipocytes sense the stiffness and secrete factors to make cancer cells secrete factors to stop their differentiation. So there may be a feedback here. Both cells are talking in real time. Or the cancer cells may be changing the stiffness, which is a dynamic process, which in turn changes the differentiation of the adipocytes. So in order to study this, we made use of our tunable, flexible engineered tissue model, where we can control the stiffness, and also we can do an experiment where we can produce conditioned media using one engineered tissue and transfer it to the other one. And we tried three different things to start with. We produced the conditioned media using the HCC cells, which is the cancer cells, on 2D in the flasks, and transferred it onto adipocyte progenitors that are encapsulated in the microvalve arrays with different stiffnesses. We in another study, we produced the conditioned media using 3D cancer cells that are grown on low or high stiffness microvalve arrays. And then we also produced conditioned media from the adipocyte and tumor co-cultures in 3D with low and high stiffness. So the first one is to test the effect of whether the cancer cell is sensing the stiffness or not. The second one is to test whether the adipocyte is sensing the stiffness or not. And the third one is to test if there's a dynamic or feedback loop interaction in between these cells. And then we started the differentiation process of the progenitors, and we started to add the conditioned media at the point that we would have seeded the tumor cells. So instead of seeding the tumor cells, we added the conditioned media from these three different conditions. So first of all, 2D conditioned media didn't do anything. So it didn't change the differentiation state, showing that the 3D spheroid formation of the tumor cells is essential for whatever they're producing to inhibit the adipogenesis. They cannot produce it when they are in a 2D monolayer form. Which clearly highlights the fact that 2D studies using tumor cells are not representative fully of their physiological processes. And then when we do the 3D conditioned media experiment, we saw that the adipogenesis, the differentiation of the progenitors to the stem, the progenitors to fully grown adipocytes are not inhibited at all by the cancer conditioned media produced in either low or high stiffness gels, only if the adipocyte progenitors are encapsulated in low stiffness. So if the adipocyte progenitors are in low stiffness microvalves, then the conditioned media coming from tumors either in low or high doesn't matter, didn't affect the differentiation state of these cells. On the other hand, if they are in the high stiffness environment, which they normally differentiate, so the stiffness alone doesn't affect their differentiation. But once we added the conditioned media from either low or high stiffness microvalves that the tumors were grown, the adipogenesis was hindered significantly. So this shows us that it is the adipocyte progenitors that are sensing the stiffness and responding to the factors that the cancer cell produces in a stiffness-dependent manner. So as a summary, we successfully established a platform for modeling the breast cancer microenvironment in the second part of the study, highlighting how we can incorporate multiple cell types of the breast and then how we can tune a factor parametrically, in which in this case it is the stiffness. And the stromal cell cancer interactions is highly dependent on ECM stiffness. So we already knew that ECM stiffness is an important factor in cancer, but this study proved that it is not only the cancer, but the stromal cell cancer interactions also are affected by this change in the extracellular matrix stiffness. And adipogenesis was inhibited in a stiffness-dependent manner, and it was the adipocytes that are sensing the changes in the extracellular matrix and then responding differently in the presence of tumor-related factors. And the 3D spheroid structure also was shown to be very essential for producing those factors, and the 2D conditioned media didn't show any effect. And as an overall conclusion and significance, I would like to highlight that aged breast ECM supports invasive metastatic phenotype, and it is alone maybe sufficient to drive tumorigenesis and or invasiveness. In the aged tissue models we develop here are time efficient, physiologically and pathologically relevant, and hold great promise for preclinical assessment and safety and efficacy trial of the diagnostic and preventative approaches, which is very much needed because, as I said, only 10% of preclinical studies that are prone to be working in the lab are translated to clinic. And the clinical importance here is aging matrix could give us a hint what may be initiating this cancer occurrence and which factors may be contributing to faster progression. And like the locks we investigated in this first study I talked about, there may be factors that are targetable in the aged tissue that can be used as preventative measures. So if you're in a risk factor for having a breast cancer, you may use this LOX siRNA as a preventative measure for treatment. Of course, this needs to be further studied in clinic, but the study highlights potential targets from the age matrix that can be used as preventative measures. At the same time, young matrix can have factors that can be used for treatment that can be identified. What's different in between young and age matrix can help identify new cancer prevention and treatment drugs. So with that, I would like to thank to all my students and postdocs and collaborators, especially Xian Zhang in biology department in Notre Dame, that we have collaborated on single cell sequencing, and Professor Sharon Stock in biochemistry that we collaborated on animal tissues. And then I would like to thank all my funding resources, especially NIH and IBAB Award that funded this project. And I would like to thank you all for listening to this talk.
Değerli bilgilerinizi ve çalışmalarınızı bize aktardığınız için çok teşekkür ederiz hocam. Dilerseniz soru cevap kısmına geçebiliriz.
Buyurun.
Hocam ilk sorumuzla başlayalım. 3B in vitro miyokard enfarktüs modellerinde hücre dışı stabilitesini sağlamak için hangi yöntemleri kullanıyorsunuz?
Tekrar alabilir miyim? Kesik geldi. İnşallah benim konuşmamda kesik gelmedi size. Şu anda internetimin açıldığını hissediyorum sizinle konuşurken.
Hocam tekrardan edeyim.
Bir soruyu tekrar edebilirseniz. Evet, lütfen.
Tamamdır.
Hayır, benden de bilir inşallah, konuşmam sırasında da kesilmedi.
Hocam konuşmanız sırasında sıkıntı yoktu. Çok güzel anlattınız. İçiniz rahat olsun o konuda.
Tamam. Yok, şu anda zor duyuyorum sizi. Kesik geliyor da onun için soruyorum. Buyurun.
Tamamdır hocam. Bir saniye bir yorumlara bakacağım. Tamamdır. Hocam 3B dediğimiz 3B in vitro miyokard enfarktüs modellerinde hücre dışı matrixin stabilitesini sağlamak için nasıl yöntemler kullanıyorsunuz? İsterseniz soruyu da geçebiliriz. Sıkıntı değil.
Evet, geçebilir miyiz? Ben çünkü hocam, benim anlattığımla ilgili olmadığını düşünüyorum.
Tamam hocam, hiç sıkıntı değil. İçiniz rahat olsun.
Bambaşka bir konuyla ilgili bir soru geldi. O yüzden anlatabilirim hocam, ama anlattığım konuyla ilgili.
Bir diğer soru. İnsan pluripotent kök hücrelerinden türetilen hücrelerdeki yaşlanma sürecini kültür ortamında nasıl takip etmektesiniz?
Farklı metotlar kullanıyoruz. Çalıştığımız en önemli konulardan bir tanesi hücre atışının kuvvetine ve atma özelliklerine bakıyoruz. Elektrokimyasına bakıyoruz hücrelerin. Tabii ki gen üretimi seviyelerine ve gen üretimi çeşitliliğine bakıyoruz. Video analizi ve mikroelektrot array'leri kullanarak elektrofizyolojilerini ölçüyoruz. Genelde bu yöntemleri kullanıyoruz.
Bir sorum daha var bu konuyla alakalı. Olgunlaşma süreleri için standart bir aralık bulunmakta mı peki?
Genelde biz bu kök hücreler, induced pluripotent stem cell'lerden oluşan hücreler hiçbir zaman şu anda tam olarak olgunlaşmış sayılmıyor zaten. Ama bizim gözlemlerimizde yaklaşık 35 gün civarında olabilecekleri en olgun noktaya ulaşıyorlar ve 35 ila 45-50 gün arasında kullanmaya çalışıyoruz bu hücreleri. Onun sonrasında çünkü yaşlanmaya geçiyorlar. Onunla ilgili çalışmalarımız da var. Onun yaşlanmalarına, hücrelerin yaşlanmasına da bakıyoruz. 55 günden sonrakine artık yaşlanmış hücre diye bakıyoruz.
Anladım hocam. Çok teşekkür ederim hocam sizin cevabınız için. Bir diğer soru. Bazı hücreler elektrik sinyallerine de duyarlı. Bu şekilde biyobilgisayar için devre elemanları üretilebilir mi? Hemen geleceğim YouTube sorusuna da.
Evet, ilk başta anlattığım konuşma tam olarak buydu. Kalp hücrelerinin elektrik sinyallerine duyarlı olmasından faydalanarak bu diyot ve osilatörleri yapıyoruz ve bilgisayar uygulamaları için kullanıyoruz.
Cevabınız için çok teşekkür ederiz hocam. YouTube'dan bir soru vardı. Hocam, çalışmalarınızda yaşlı mikroçevrede siferoidlerin kaybolması neden kaynaklanmış olabilir?
Bizim anlattığım gibi tahmin ettiğimiz hücre yüzeyindeki kaderin ekspresyonunun hücre içine alınmış olması. Bunu bütün konuşmayı detaylı olarak tekrar anlatmam gerekir. Evet.
Cevabınız için çok teşekkür ederiz hocam. Bir diğer sorumuz, yaşlı ve genç kalp dokusu mikroortamı arasındaki farklar nelerdir? Kardiyovasküler hastalıklar açısından önemi nedir?
Çok gene kalp durumunda da meme dokusu için anlattığım gibi çok farklı değişen şeyler var. Değişen şeylerden en gene etkili olanlarından bir tanesi kompozisyon. Bu extracellular matrix içinde bulunan farklı proteinler var. Bu proteinlerin oranları değişiyor ve onun haricinde extracellular matrix'in içinde olan sitokinler var. Bunlar daha aktif protein molekülleri. Bunların da çeşitleri ve miktarları değişiyor. Bunların hepsi katkıda bulunuyor yaşlanmış dokudaki görülen geç iyileşmeye.
Cevabınız için çok teşekkür ederim.
Ve şeyi de değişiyor, sertliği de değişiyor. Kalp dokusu yaşlandıkça çok sertleşiyor genç dokuya kıyasla.
Anladım hocam. Cevabınız için çok teşekkür ederiz. Bir diğer sorumuz YouTube'dan geldi. 29 genin indüklendiğinin fark edildiği deneyde sağlıklı yaşlı hücrelerde mi çalışılmıştı yoksa kanserli ve yaşlı olanlarda mı?
Sağlıklı ve genç hücrelerde. Bu hücrelerin hepsi sağlıklı ve normal hücreler. Değişen tek şey extracellular matrix'in yaşı. O 29 genin indüklendiği çalışmada sağlıklı insan epitel hücreleri.
Cevabınız için çok teşekkür ederiz hocam. Bir diğer sorumuz. Yaşlanma mekanizmalarının zamana bağlı hücresel hasar birikimlerinden kaynaklandığı düşünüldüğünde günümüz şartlarının geleneksel yaşlanma prosesi ve modellemesinde yarattığı zorluklar nelerdir?
Valla dediğim gibi biz bunun hücre içindeki değişimlerin ötesine bakmaya çalışıyoruz. Bu konuda sırf yaşlılık üzerine de çalışmalarımız var. Basılmış makaleler, ilgilenen arkadaşlar benim ismimle bakarlarsa bulabilirler. Hücrenin içinde değişen şeyler hücreler arasındaki iletişimi değiştirdiği için hücreler arasındaki iletişim eksikliğinden dolayı gelişen yaşlanma ile ilgili sıkıntılar olduğunu düşünüyoruz. Hem hücre arasındaki ilişkilerin azalması hem de hücre ve matris arasındaki ilişkilerin değişmesi, ikisi de hücre içindeki değişimler kadar önemli ve şu ana kadar göz ardı edilmiş iki tane olgu. Bunların üzerine yoğunlaşıyoruz benim laboratuvarımda.
Cevabınız için çok teşekkür ederiz hocam. Bir diğer soru. Mikro ölçekli doku mühendisliği uygulamalarında iskele mimarisinin tasarlanmasında dikkat edilmesi gereken noktalar nelerdir?
En önemli şeylerden bir tanesi taklit etmek istediğiniz ya da çalışmak istediğiniz dokuya uygun olacak şekilde, benzeyecek şekilde bir iskele ya da yapı oluşturmak. Ve de dediğim gibi bu dokuların en önemli faydalarından bir tanesi değişik parametreleri birbirinden ayırarak çalışmaya elveriş vermesi. O yüzden de geri kalan parametreleri nötr olacak şekilde tasarlayıp sadece çalışmak istediğiniz parametreyi değiştirecek şekilde bir dizayn yapmanın faydası olacağını düşünüyorum. Her şeyi bir anda değiştirmek yerine.
Cevabınız için çok teşekkür ederiz hocam. Bir diğer soru. Kalp kapakçığı mikroortamına uygun bir hidrojel yapısı nasıl tasarlanmalıdır?
Kalp kapakçığı ile ilgili benim laboratuvarımda çok çalışmıyoruz ama hyaluronik asit içermesi gerektiğini düşünüyorum. Ama o benim çalıştığım bir alan değil. O yüzden çok fazla konuşmak da istemiyorum üzerine.
Cevabınız için çok teşekkür ederiz hocam. Diğer bir soruyla devam etmek istiyorum. Günümüzde canlı dokunun doğal mikro çevresi ne kadar başarılı taklit edilmektedir ve geliştirilmesi gereken yönler nelerdir?
Dokusuna göre değişiyor ama evet daha çok yolumuz var. Tabii ki aynısı gibi doğal dokunun aynısı gibi olmayabiliyor. Ama burada unutulmaması gereken az önceki soruya cevap olarak da söylediğim doğal dokudaki her şeyin her parametrenin aynısını yapmaya çalışmak değil zaten amaç olmamalı da. Bu hastalık modellerindeki amaç farklı parametreleri çalışabilmek. O yüzden de mesela Stephenson etkisine konuşmayacağım İngilizce Türkçe dedim ama yapamıyorum. Çok geriliyorum o yüzden de. Dokuların sertliğinin etkisine bakacaksak eğer mesela onu mümkün mertebe benzer şekilde taklit etmek önemli. Bunu başarabilen dokular var. Dokuların şekilleri yani yapılmış çalışmalar var. Dokuların şekline benzer şekilde taklit etmenin de benzer şekilde önemi var. Dediğim gibi kompozisyon ve structure ya da overall pattern'ı güzel bir şekilde yapabilmek önemli olan ve o noktaya doğru ilerleyen çalışmalar var.
Cevabınız için çok teşekkür ederiz hocam. Bir diğer soru. Genç hücreleri yaşlandırılması ile Alzheimer gibi hastalıkların erken tanısını ışık tutacak çalışmaların gerçekleştirilmesi ne derece mümkündür?
Evet, kesinlikle çalışılan bir alan olduğunu düşünüyorum ve bu yaşlı doku modelleri konusunda daha da çalışmalar yapıldıkça biz bunu şu ana kadar yaptık, meme kanseri ve kalp için yaptık ama eminim ki başka laboratuvarlarda umuyorum ki benzer modelleri Alzheimer ve benzeri gene yaşlı hastalarda sıklıkla görülen hastalıklar için benzer şekilde devam edecektir çalışmaya ve faydası olacağını düşünüyorum çok.
Cevabınız için çok teşekkür ederiz hocam.
Bir
diğer soru. Yaşlanma sürecindeki bireysel ve çevresel faktörler doku mühendisliğinde ne kadar zorluk oluşturmaktadır?
Çok güzel bir soru. Biz bu doku modellerine başladığımızda onun için fare dokusuyla başladık. Fakat geçtiğimiz bir senedir insan dokularıyla da çalışıyoruz. Genç ve yaşlı insan meme dokularıyla. Tabii ki fare dokusunda çevresel ve bireysel farklılıklar çok kontrollü. Çünkü fareler laboratuvar ortamında tutuluyor ve benzer şekillerde üretildiği için oradaki farklılıklar daha az. İnsan dokusuyla çalışmaya başladığınızda tabii ki bunu faktöre almak lazım. Ama genelde yapılan şey daha çok sayıda örnek kullanarak işte 3 ila 4 örnekten 12-15 örneğe çıkararak farklı örnek sayısını buna bakmaya devam ediyoruz, çalışıyoruz ve bunları yani göz önüne almanın en yapılabilecek yolu baktığınız örnek sayısını dediğim gibi arttırıp popülasyon içinden daha fazla örnekleme yaparak yapmaya çalışıyoruz. Bu aradaki farkları ortalamasını almaya çalışıyoruz büyük ölçüde.
Cevabınız için çok teşekkür ederiz hocam. Bir diğer soru. Doku kültürleri ile mikro akışkan teknikler hastalık fenotiplerinin incelenmesi için combine edilebilir mi?
Evet, evet ediliyor zaten. Biz de ediyoruz, başka laboratuvarlar da ediyor. Konuşmanın başında gösterdiğim bir organ prezervasyon solüsyonu çalışmamız vardı. Orada mesela yaptığımız mikro akışkan çiplerin içinde yaratıp bu kalp dokularını akışın etkisine de bakabiliyorsunuz ve kalp krizi için de benzer şekilde çalışmalarımız var. Akışı durdurup kalp krizindeki kan akışının durup sonra tekrar damar açıldığında akışın devamını modellemek için kullanıyoruz biz ama başka laboratuvarlarda da başka amaçlarla çok kullanılıyor. En büyük faydasından biri bu mikro akışkan sistemlerin ilaç etkilerine bakarken çok faydalı oluyor. Çünkü ilaçların dozajını ya da işte lokalizasyonunu, dokunun neresine vereceğinizi vesaire kontrol edebiliyorsunuz.
Çok teşekkür ederiz hocam cevabınız için. Son sorumuz.
Tamam.
Doku yaşlanması yanında hücre dışı bir matrix yaşlanmasını da incelemek faydalı mıdır? Ve bu tür yaklaşımlar günümüzde bulunmakta mıdır? Tekrar edeyim mi?
Hayır da doku yaşlanması derken zaten baktığımız şey ekstraselüler matrix yaşlanması. Yani orada aynı şey diye düşünüyorum. Dediğim gibi hücre açısından bakanlar var. Başka bir nüans vardıysa soruda ben anlayamadım belki ama bizim doku yaşlanması olarak baktığımız, daha doğrusu odaklandığımız kısmı, doku yaşlanmasının odaklandığımız kısmı ekstraselüler matrixin yaşlanması zaten.
Anladım hocam. Cevabınız için çok teşekkür ederiz.
Rica ederim.
Katılımınız ve verdiğiniz değerli bilgiler için tekrardan çok teşekkür ederiz.
Rica ederim.
Sizi aramızda görmekten dolayı çok mutluluk duyduk.
Ben çok teşekkür ederim.
Dileriz.
Çok teşekkürler davetiniz için ve sorular için. Hoşça kalın.
Sağ olun hocam. Görüşmek dileğiyle tekrardan.
Hoşça kalın.